Welcome back, everyone. We are on our final packet for this module, and then we'll have the exam on everything we've learned so far. So we are on to cellular respiration. I'm totally pumped. I love this. It's somewhat hairy and complicated, but I'll try to make it as simple as possible for you guys. And let's go. So, your textbook reading is here, review questions here, key concepts, key terminology as always. And then our question of the day, when you lose weight, where does it go? So here we have a molecule, and just by looking at it, we can see it's a hydrocarbon. Hydrogen's carbons. It's a fatty acid. And what we're going to do, my Sharpie never wants to work at first, is this carbon backbone is going to be broken down. We'll come back to that. So on to the next page. On the pre-lecture assignment, I am going to cover it this time just because it's background information. I want to make sure you understand. And look at that. I finally got colored copies. Yay. So let's start filling in the blanks. Metabolism is the sum of all the chemical reactions that occur within the body. And reactions such as cellular respiration, which we're going to go over, often involve metabolic pathways. And when you hear of metabolic pathways, that's just when we take one molecule, turn it into another, turn it into another, and so forth. And all of these actions, these reactions require enzymes to facilitate them. So I kind of answered the next question. Each reaction in a metabolic pathway requires a specific enzyme. We've talked a little bit about enzymes before, but they are proteins that facilitate, speed up chemical reactions in the body. Keep getting ahead of myself. Enzymes are metabolic assistants that speed up the rate of a chemical reaction. So let's zoom in on this picture here. Hopefully that will focus. So this has two terms, degradation and synthesis. We're going to call these, instead, the terms that we learned, hydrolysis. So remember, if you break a molecule apart, that's called hydrolysis. And building, or synthesis, we have those dehydration reactions. Alright, so let's first look at hydrolysis. So you will have a substrate, is what we call it, before the enzyme interacts with it, and it attaches to the active site. And enzymes are picky, they're choosy about what substrates they'll react with, and if you'll look here, it kind of fits in there like a little puzzle piece. So the substrate will come and bind with the active site, and then this enzyme is breaking this product down, or breaking this substrate down into smaller products. You can see it's broken them down into two smaller products, and then the enzyme can carry on to do it again and repeat this process over and over. So enzymes are reusable. Dehydration, again, is the building. So now if we take two substrates, they just fit perfectly together in the enzyme's active site. And the enzyme substrate complex is what we call them when they're together here. And they will bind those two together. And now you have the product. And again, that enzyme can carry on its way to do what it needs to do. Again and again. Oh shoot, I just remembered. I brought I brought my whiteboard home from school so I could write on it, and I left it downstairs, so give me like 30 seconds while I go grab it. Okay, now I'm gonna be out of breath. I borrowed this from my school. I have my whiteboard, I have my cute little eraser, and my little expo markers so I can draw pictures for you guys. So that's what I'm gonna do right now. You don't necessarily need to write this down anywhere, but just to show how enzymes work, we were to draw a little table here, and this y-axis represents energy. In order for chemical reactions to happen, it requires the input of energy. So it'll go something like this. And then down here we'll have our products. This point right here is when we've got enough energy for the reaction to occur, and we call that the activation energy. Which is just like it sounds, it's the amount of energy needed to activate the reaction. So what the enzymes do is they lower that activation energy. Okay, so now it's only going to take this much energy for that reaction to occur. Get out of the glare there, as opposed to this much. So enzymes lower the activation energy. Please tell me this erases. Oh, yay! How perfect. Okay. It's the little things, my friends. All right, so knowing that, let's read through this number 2 here. Circle the correct term in the parentheses. Use the diagrams at the top right to help you. So, enzymes are proteins that catalyze, that word means speed up and help, reactions in which molecules are combined or broken down. So the things that enter, if we look back at this picture, are the substrates. They enter, looking back at the picture, this is called the active site where they bind. I mean, activation chamber, that's a cool word, but it's active site. On the enzyme, and the enzyme holds the substrates in such a way that they are forced to either break apart or combine. The resulting products, that's our end products, I can't think of another word, are then released and the enzyme is ready to go to be used again. So like we saw on the whiteboard picture, an enzyme's active site only fits a specific substrate like puzzle pieces. And this comes back to why the shape of proteins are so important. If you remember our protein folding activity, if your body is making these enzymes and they're not the right shape, they can't grab and bind onto the substrates that they need and can't catalyze these reactions, and it can have devastating results. So the shape of the enzyme is very important, so that those substrates can fit on the active site. And then mine printed funny. I don't know if yours looks like this, but the energy must be added to cause that to cause molecules to react with one another is called the energy of activation. Sorry, I don't know what happened there. And by lowering this energy, the enzyme increases the rate of, I'm guessing that says the reaction. Okay, so metabolism, enzymes. Now let's look at the glorious mitochondria. So we're going to label this, and it is important because in these reactions you need to know where they are happening within the cell and also within the mitochondria. So this right here, this outer line is called the outer membrane. All of the inside of the mitochondria is called the matrix. It's a cool name, cool movie. This line right here is the inner membrane. And then the space here in between the outer membrane and the inner membrane is called the intermembrane space. So inter means between, so that's the space between the membranes. Okay, before we start into cellular respiration, I've got these written down on this cute purple paper. So questions that we'll be able to answer after today. So how does my body turn food molecules I eat into energy? We know we have to eat to get energy, but how does this happen? Where does weight go when I lose it? I mean, that's matter that's being lost. Where does it go? Where does oxygen go that I breathe in? Where does CO2 come from that I breathe out? I think we all know we exhale CO2. Where does it come from? And why do I die when I stop breathing? So cellular respiration is going to give us these answers. Behold, cellular respiration has the answers. I've written out the reaction. I don't think this is necessarily something that you have to memorize, but as you go through and learn this, you'll see where this comes from. So what goes into the reaction is glucose and oxygen, and what comes out is CO2, water, and ATP. So remember, ATP is that energy currency of the cell that the cell needs in order to do the work it's got to do. It can't use glucose. Glucose has to be converted, if you will, to ATP. All right. Cellular respiration. Okay, so this is how your body harvests energy from food. So the reason you eat is because you need those glucose molecules and sometimes others, as you'll see, to be able to break them down to form ATP so your cells can do work. So why you eat. So when we go over the cellular respiration, we're typically looking at carbs. But keep in mind that lipids and proteins can do this as well. So they can all undergo cellular respiration. To make ATP. So let's zoom in on this diagram here. So when you eat something like a carbohydrate, remember that's a polymer, a macromolecule made up of a bunch of little monomers, and a lot of times those monomers are glucose. And your digestive system needs to break those polymers down into their individual subunits so they can be absorbed into your blood, carried throughout your body, and then in the capillaries, they can start to move into cells. So in this picture, we've got our phospholipid bilayer, and glucose is too big to just diffuse through it. It doesn't look like it in this picture, but typically we're going from high glucose out here to low. So that's simple diffusion, high to low. But since it can't go through the membrane, it needs this helper, this protein helper, to help it get inside the cell. So remember, we call that facilitated diffusion. It's going from high to low, but it can't simply just get through the phospholipid bilayer, so it needs this protein helper. I'll just write that up here. Facilitated diffusion. So now, we've got glucose inside of the cell. Now we're ready to begin the process to make ATP. Alright, mitochondria. This is the classic definition. It's the powerhouse of the cell. And it's an organelle. So we talked about organelles last time, like the nucleus, the endoplasmic reticulum, the Golgi apparatus, the microtubules, all of those things. The mitochondria is another organelle. We just skipped talking about it because it's so awesome, it needs a whole lecture on its own. So this is where cellular respiration is carried out. So it carries out cellular respiration. And most, not all, but most of the process of cellular respiration occurs in the matrix, in this middle portion of the mitochondria. It also occurs, a certain part of it, and you'll see it occurs along the inner membrane. So if you look here, the inner membrane is highly folded. It has all these projections. And so what is the advantage to those many folds? If you ever have something with a lot of folds, it's usually to create more surface area. Alright, so there's part of the reaction that's going to occur along this inner membrane. And with all these folds, that gives us lots of space for these reactions to happen. Alright, and one more bullet point here. The mitochondria generates ATP. which is like a rechargeable battery. So we did talk about ATP, but just this is a very simplified model of it. It stands for adenosine triphosphate. So you've got an adenosine here, and you've got 3 phosphates, triphosphate, bonded to it. And this bond right here is a high energy bond. And I don't know if you guys ever watch any of Hank Green's videos. I love that guy. But he described this as 3 kids squished on a school bus seat together, and 2 of the kids can't stand this kid on the end. And so they're just going to want to pop him out of there. And when that bond breaks, when they push him out of there, it releases a lot of energy, and your cells can harvest that energy to do work. So when we say ATP is like a rechargeable battery, so we have ATP, and that gets broken down into ADP, so adenosine diphosphate, so we're cutting off this last one here, plus the phosphate that you removed. And doing that, energy from ATP breakdown is used for metabolic work. Then these parts are going to be used again to make ATP using the energy from cellular respiration. So this goes in a cycle. There are 2 types of cellular respiration. There's aerobic, which requires oxygen, and anaerobic, and anytime you see AN in front of something, that usually means without. So anaerobic does not require oxygen. Alright, so know that there are 2 types, aerobic and anaerobic. If it's aerobic, it's needing oxygen. If it's anaerobic, it can happen in the absence of oxygen. That's just a scratch on my desk, by the way. Alright, cellular respiration occurs in 4 steps. Glycolysis, pyruvate oxidation, the citric acid cycle, sometimes called the Krebs cycle, and the electron transport chain. And there's a lot going on here. And we're going to keep it, again, as simple as possible, but knowing as much as you need to know. So for each of those 4 steps, you need to know where it occurs in the cell. And just so you know, I think 3 out of these 4 occur in the mitochondria. So if you're asked where it happens and you're not sure, and you're going to go with a guess, go with mitochondria. But hopefully you can memorize these. You need to know what molecules go into each reaction and what the products are, what molecules come out. Now, at the very end of this, we're going to come to this page and summarize everything. And this is going to kind of put it all nicely together on one page for you. But again, we'll come back to that at the end. But it's going to show you where it happens in the cell, what molecules go in, and what molecules come out. So, for explaining this today, we're going to follow the path metabolism of a single glucose molecule, although we mentioned that fats, proteins, and other carbs are also metabolized during cellular respiration. So if we look at how many glucose or how many carbons are in a single molecule of glucose, its formula is C6H12O6. So we've got 6 carbons. And its molecular structure looks like this. It's like an O. Like we saw with the monomers, it's a whole bunch of these stuck together to make the polymers. But to make it easier to see what's happening to these 6 carbons, we just kind of unravel it and show it in a line like this. And I even simplify it even more. And glucose is 2, 3, 4, 5, 6 carbons. All right. So if I were to define cellular respiration in one sentence, I would say we are going to break down these bonds between the carbons and take that energy, because every time you break a bond, it releases energy to make ATP. Obviously, there's a lot more going on than that, but if I were to summarize it, it's breaking these bonds, using that energy from breaking those bonds to make ATP. All right, so we're going to focus on this model as we go through cellular respiration, because it's easier to see what's going on with the carbons. Okay, step 1, are we ready? Glycolysis. When you see lysis, what does that make you think of? Like hydrolysis. It's breaking down. So this means the breaking down of glucose. So where does this happen? One of our 3 important questions. This occurs in the cytoplasm of the cell. And remember, the cytoplasm is just that jelly-like fluid. It's peach in this picture that all of the organelles are kind of suspended in. It's anaerobic, so it doesn't require oxygen. And what happens is glucose is broken into 2 pyruvates. So glucose comes in, 2 pyruvates come out that are 3 carbons each. Then the pyruvates move into the matrix of the mitochondria. All right, so we've got glucose. In glycolysis, in the cytoplasm of the cell, we're going to break this bond right here. And what that gives us is 2 3-carbon molecules called pyruvates. Now those pyruvates are ready to head into mitochondria for the rest of these reactions to happen. So the yield, meaning what molecules come out, our 3rd question, is we end up with 2 pyruvates. Remember, that's 3 carbons. We get 2 ATP. So we do get a little bit of energy from this reaction, but not a lot. And we get something called NADH. You know you're in Utah when heck is included in your lecture notes. What the heck is NADH? What it is, it's a molecule, and it's an electron carrier. So what happens is NAD plus picks up 2 electrons. I'm going to abbreviate electrons like that. Hopefully that's okay. And 1 hydrogen ion. During glycolysis, to become NADH. NADH will later dump off this hydrogen and electrons that it picked up to help make ATP. So right now you're probably thinking, why does this matter? It matters. And you're going to see when we get to the last step, the electron transport chain, that this NADH carrying these protons and electrons are going to become very important. So NADH will later dump off its hydrogen and electrons to help make ATP. So I'm going to write this up here. This is just showing it picking up those hydrogen ions and electrons. And by the way, when I say hydrogen ion and proton, they're the same thing because a hydrogen atom only has one proton and one electron. And when it loses that electron and gets a negative charge, all that's left is a proton. So protons and hydrogen ions mean the same thing. I should stay consistent with what I say, but yeah, sorry if that's confusing. So you've got NAD+, and that picks up the hydrogen ions and 2 electrons, and that gives us NADH. So moral of the story, NADH is picking up after this carbon glucose molecule is breaking into 2. A result of that, you've got some hydrogen ions and electrons, and it's going to grab onto those and hold on to them for later, because we're going to need them. Alright, that was step 1. You good so far? Everyone say yes, even though I can't hear you. Okay, next we have step 2, pyruvate oxidation. So one of our big 3 questions is, where does this happen? This occurs in the matrix of the mitochondria. So in that center part of the mitochondria. And what's going in are those 2 3-carbon pyruvates that were yielded in glycolysis. And they'll be broken down into 2 2-carbon molecules called acetyl-CoA. Alright, so what's going in are those 2 3-carbon molecules we made in glycolysis. We're going to break them down even further. We're going to cut off another carbon from each of those, and we're going to be left with 2 2-carbon, and the name of that molecule is acetyl-CoA. So the yield, what do we get from this? We get 2 acetyl-CoAs, those 2 carbons like that. We have 2 of those. We get a little more NADH. So as a result of those carbon-carbon bonds breaking, we're going to have some electrons, we're going to have some hydrogen ions. The NADH is going to grab it, hold on to it, and save it for later. And those 2 carbons that were broken off right here, that's CO2. So to answer one of our questions here. Where does CO2 come from that I breathe out? It comes from some of it comes from these carbons breaking off and being released as essentially a waste product, a byproduct of CO2. Your cell gets rid of it, it goes into your bloodstream, and you breathe it out. So we'll make sure we know that. These two carbons came from the carbons that were broken off pyruvate. So if ATP is abundant, we've been eating lots of sugars, we have lots of ATP. At this point, the acetyl-CoA is converted to triglycerides. So we've got lots of ATP we don't need anymore, but we don't want it to go to waste. So your body will convert it to triglycerides and store it as fat. In case we need that energy later. So can you get fat from eating sugar? And if so, how? So we'll write the answer over here. Excess food is not metabolized all the way if no ATP is needed. Instead, the excess food is stored as fat. That's kind of repetitive, but it's okay. All right, step 3, here we are. The glorious citric acid, or the Krebs cycle, sometimes called the Krebs cycle because a man named Hans Krebs actually figured out this reaction, which is quite incredible. And we call it the citric acid cycle because that's what we're starting with here. So this is kind of the bane of every biology student's existence, is the citric acid cycle or the Krebs cycle. And as a student, I would hear the teacher, I don't want to admit this, I would hear them say citric acid or Krebs cycle, and I would immediately zone out, tune it out, because I'm like, this is too much. There ain't no way that this brain is going to understand all this. And I graduated with a degree in biology, don't tell anyone, without understanding the Krebs cycle. But then guess what? I came back to what was Dixie State at the time, and I had to teach biology classes, and guess what? You can't teach it unless you know it. So I had to learn it, and after I learned it, I was like, you know what? That's not that bad. It's not that bad. And you guys, especially for this class, don't have to know it in a whole lot of detail. So be thankful for that. But anyway, one of our big questions is, where does this happen? Again, this occurs in the matrix of the mitochondria. And here, all the remaining carbons are given off as CO2. So again, this is just waste product, so your cell's going to get rid of it. It's going to diffuse out of your cells into the blood. You take into your lungs where you exhale it. And the yield, what are we going to get out of this cycle? We're going to get a little bit of ATP. We're going to get some more NADH, carrying those hydrogen ions and electrons. We're going to get something called FADH2. Well, what the heck is that? Basically, it does the same thing as NADH. It's just picking up some hydrogen ions and electrons. It's going to carry them to where they need to go, and we're going to get rid of 4 CO2 molecules. So at this point, at the end of the Krebs cycle, the carbons of the original glucose molecule have all been used to make CO2 that you exhale. So what happens, and again, be glad you don't need to know all these steps here because there's a lot going on here. But what is getting put in are these acetic acids that we ended with after pyruvate oxidation. And they're going to help form something called citric acid. That's why it's called the citric acid cycle. We're also going to put in some ADP and phosphate. This is going to end up making ATP, some energy. These are going to end up being CO2. And then we're going to have these guys pick up some hydrogen ions and electrons. Alright? And then the process starts all over again. Okay? Can't believe that's all I have to tell you about the citric acid cycle. So we've already answered this question. What is the status of the carbons after the citric acid cycle? We've used them all up, and they've all been exhaled from as CO2. So the CO2 you exhale comes from that original glucose molecule. So that answers another one of our questions. Where does weight go when I lose it? You exhale it. You breathe it out. Isn't that crazy? So it's the carbons in your CO2 that you're breathing out came from the carbs or the triglycerides in your body. Pretty wild. Okay. I hope you're still with me. Our fourth and final step. This is where the big money is made. The electron transport chain. Alright, this is where the majority of our ATP is made. So far we've made a little bit. Not a whole lot. And this is aerobic. So we need to have oxygen. And the electron transport chain is a chain of proton pumps. Remember, proton is just another word for this. And it's the active transport of hydrogen ions from the matrix to the intermembrane space. This is where NADH and FADH2 become important because they're going to donate all of these hydrogens, all these protons and electrons they've been carrying to this reaction. They're going to carry them over there, and it allows this to happen. So hydrogen ions and electrons are donated from NADH and FADH2. We need oxygen as a final electron receptor for this reaction. It's going to clear electrons, pull them out from the electron transport chain so this thing can keep going. Okay, without electrons or oxygen, all these electrons, everything would get backed up. You wouldn't be able to make any more ATP. And then guess what? You would die. So final electron receptor acceptor combines with hydrogen ion to become water. Alright, so that's where the water, the reaction comes from. So where does the oxygen gas that you breathe go? So it's going to go from your lungs, diffuse into your bloodstream. Your blood's going to carry it to the cells, into the capillaries, and then it will diffuse inside of your cell. You see it right here. And it has to be there to grab the electrons that are going through the series of proteins here is called the electron transport chain, so we can keep it flowing. Okay? So without that, you can't make any more ATP. Your cells can't do work, and you die. So that answers our question here. Why do I die when I stop breathing? You don't have oxygen here to pull the electrons out of the electron transport chain to make water. Can't make any more ATP. Your cells lose energy, can't function, and you die. And we're going to talk about how this works in a second. So what happens if you stop breathing? If no oxygen is at the end, to accept electrons, the pumps get backlogged, and no ATP is made. And this means you die. Not trying to be dramatic or anything, but it is what it is. Alright, so let's see if I can explain this picture here to you guys. Because this is where all your ATP is coming from. Not all of it, most of it. So we have zoomed in here on the intermitochondrial membrane, and we have these series of proteins called the electron transport chain, and then we've got the matrix here, and then the intermembrane spaces here. So what's going to happen is NADH and FADH2 are going to come drop off their electrons, And this yellow line here represents the electron flow. And the flow of the electrons from these pumps as they're getting pulled through and picked up by oxygen causes these protons or hydrogen ions to be pumped against their gradient outside into the space, the intermembrane space. Alright, so I'm going to repeat that one more time. We've got NADH and FADH2, and they're coming and they're dropping off those electrons and hydrogen ions they've been carrying. The electrons are going to move down the electron transport chain, combine with oxygen to make water. And as that happens, this causes the hydrogen ions to be pumped out into that space between the membranes. So what you're left with is a gradient. Okay, and kind of think of it as potential energy. So you've got all these hydrogen ions out here, a high concentration. You've got a lower concentration here. And so naturally, they're going to want to diffuse from high to low. And so think of it as like water built up behind a dam. So here's this picture down here. And I don't know much about this, but think of the hydrogens as being all over here in that intermembrane space. Got a whole bunch of them. And if they're allowed to flow, you're going to have all this energy coming through here. It's going to turn this turbine. So we're going to think of this as ATP synthase, which we'll look at in a second. And then ATP is what comes out. So, ATP synthase is an enzyme. Anything that ends in ASE is typically an enzyme. And what it does is pumps those hydrogen ions from the intermembrane space back into the matrix. So from high to low concentration. And this makes ATP. Remember, it's an enzyme. So it's going to take the substrates, ADP and a phosphate, and squeeze them together to make ATP. So this makes ATP by combining ADP with a phosphate. Alright, so let's add this here, ATP synthase is just like a hydroelectric dam. The water turning the turbine produces energy. Sorry, my writing's getting sloppy. I hope you can read that. I'll read it one more time. ATP synthase is just like a hydroelectric dam. So the water turning the turbine produces energy. So again, we have all these hydrogen ions that were pumped into the intermembrane space, creates a gradient, high to low, and through the ATP synthase enzyme protein pump, They'll flow from high to low. This would be like the reaction site here. And this combines ADP with the phosphate to give you ATP, to make some energy. Alright, so we have another picture of this here. So this is the end of the electron transport chain. The electrons are coming out. Oxygen is picking them up, grabbing two hydrogen ions to make water. That's where water comes from in the reaction. And again, those protons, those hydrogen ions have been pumped out. So we've got a high concentration out here, a low concentration in here. And they're going to diffuse through this ATP synthase. And it actually causes it to turn. I think the actual molecule turns. And this brings and makes these two things stick together to make a whole bunch of ATP. And the amount of ATP that's made is debated in every textbook. It'll say something different, but I'm going to say it makes about 30-something ATP. Alright, so this electron transport chain is where the bulk of your ATP comes from. We can really use that energy and this enzyme to stick a whole bunch of these together and make a whole bunch of ATP. Alright, back to our question of the day. You guys okay? We're almost hitting an hour. But like I said, my other lecture went a little shorter, so we're okay. So again, we kind of talked about this, but you breathe it out. When you lose weight, where does it go? You breathe it out. So fat is mostly a carbon backbone. Like we looked like at the very start, that carbon backbone. These carbons enter cellular respiration. And are given off as CO2. Pretty cool. To be honest, I don't really know what this picture means, but you can look at it and see if you can figure it out. I just know that the carbons in those fats through cellular respiration are broken down into the one carbon CO2 molecules. You breathe them out. Alright, we are going to come back in just a second and summarize all of those four processes. But first we need to cover fermentation. So this is our anaerobic respiration. So fermentation kicks in temporarily in absence of or very low oxygen. So remember, when there's not enough oxygen, your cell can't do that last part. The electron transport chain, where the majority of our ATP was made. And so your cells will continue to produce that small amount of ATP through glycolysis And per glucose molecule, it only makes 2 ATP. So is this very efficient? No, you're not getting the biggest bang for your buck. You're only getting 2 ATP for your glucose as opposed to like 38 or something with cellular respiration. So I'm not really going to get into this process, but just know that fermentation kicks in and that it's just temporary and it only yields a couple of ATP. Okay. Here we go. Let's summarize this. Can we do it? This chart is going to be a great thing to study. Let's see, zoom out a little bit. All right, so to start this process, we have glucose. And we represent it as a 6-carbon chain. The location, glucose, enters the cell from the blood. Okay, your blood picked it up from the digestive system. It enters into the cell through facilitated diffusion. And now that we're in the cell, the cytoplasm, we can do glycolysis. Remember, that means the breaking up of glucose. So we're going to take that 6-carbon chain. We're going to break it in 2. We're going to end up with 2 3 carbons. And we call these pyruvates. Okay, glycolysis happens in the cell cytoplasm. And what it yields is just the energy from that bond breaking. We're going to get 2 ATP from that. We're going to get the 2 pyruvates that you see. And we're going to get some NADH. That is that hydrogen ion and electron carrier that's going to take that in the end to the electron transport chain. All right, our next step. These 2 pyruvates are going to head into pyruvate oxidation. So what's going to happen is we're going to cut off a carbon from each of these. So you're going to end up with 2 carbons from each of these. And these are what we call acetyl-CoA. This happens in the matrix of the mitochondria. And we end up with the 2 acetyl-CoAs. Some more NADH that picked up some hydrogen ions and electrons when that other bond was broken. And then those 2 carbons that broke off are given off as carbon dioxide, as CO2. All right, now those 2 acetyl-CoAs are going to head into our citric acid cycle. Where all remaining carbons are lost as CO2. This also happens in the matrix of the mitochondria. And we're going to get some more NADH. And now our FADH2. Again, those just picked up hydrogen ions and electrons as a result of those bonds breaking. The remaining 4 carbons that we have are going to be released as CO2. And we're also going to get a little bit of ATP out of this. And now we're going to head to the electron transport chain, the 4th and final step. So this is where all the NADH and all the FADH2s that we've yielded from these other reactions are going to come over. They're going to drop off those electrons and protons. And then remember, this is aerobic, so we need oxygen gas to pull those electrons out so the chain can keep going. This happens in the matrix of the mitochondria. This is going to get us about 32 ATP. And when that oxygen gas pulls the electrons out, it's going to give us some water. So the end result of this is ATP and water. And this happens across the inner membrane in that electron transport chain. And the intermembrane space where all of those hydrogen ions go. Okay. We did it. We did it. I want to know how you guys are feeling. Okay, I have this finished filming a little in advance of when you will actually be taking your first exam. I'm still not quite sure how all of that works, so watch for an announcement from me, probably, for like maybe the best way to study and just reminders of how and where you can take your test. But yeah, this is the end of what's going to be on that first module test. So good work. You guys officially know what cellular respiration is, hopefully. And we'll see you in the next module.